What's going on, sentient microbial sacks? As you may have heard, we're currently in a global pandemic that's affecting everyone, even scientists. Just about all college campuses are closed, and researchers are either prevented or advised against being in the lab. So, is there any research getting done? Well, of course. A lot of researchers are focusing on data analysis and writing this time, but I'm very thankful because I actually get to do my lab work at home. I work in an invertebrate paleontology lab, which means I dig up cute little creatures called foraminifera or forams. Forams are single-celled marine organisms that reside inside a shell that we call a test. There are two major groups of forams: benthic forams, which live on the seafloor, and planktonic forams, which float at the surface of the ocean, similarly to plankton and cyanobacteria. The majority of these organisms secrete calcareous shells from the calcium and carbonate ions present in the ocean. As a result, the tests are buried in sediment after the organisms have died and can remain there for millions and millions of years, similar to dinosaur bones. Since forams are small and have been present in every ocean on Earth for millions and millions of years, they are used by geologists to date rocks, a process known as biostratigraphy, used as a proxy for ancient ocean conditions, a very important part of reconstructing the climates of past or paleoclimates, and to study the evolution and ecology of these organisms over time to better understand how changing climate affected the life of organisms that are highly dependent on their environment for survival. All of that is to say, forams are really interesting organisms. But how do we get them out of sediment to study them? Scientists go on research vessels to oceans all over the world and carefully collect cores of sediment deposited at any time from the present day to hundreds of millions of years ago. These cores are split into depth intervals, which are further split into little vials that are picked by scientists like me. And when I say picked, I mean I suspend the sediment in water, look at them under a microscope, collect the forms of interest with a very small brush, and put the specimen onto special slides that allow me to catalog them later. This is what forams look like on a slide under a standard magnifying glass. If you look closely, there's multiple little specks of white. There are different sizes, and those are the creatures of interest. Here's what they look like under the microscope. My current project is about understanding the paleoecology of the region this core was collected in. So I have to pick several hundred forams from every depth interval, which means I've got a lot of time to spend on the microscope. Lucky for me, I enjoy the work, and also I have no choice but to be inside for the majority of the day. And there you have it, a look into what lab work can look like in these very strange times. I hope you've enjoyed this video. If you have, please give the video a like, comment with any questions you have for me, and subscribe for more science content. As always, keep it loopy.